So welcome back, guys, for the Sync in English. And last videos we talked about brain structure and their functions, etc. And in this video, we'll try to understand how actually the brain images were taken and how we can study the brain activity or functions. So let's get started. Yeah. So here, we'll be talking about different methods that we use. So commonly used techniques recording, which records the brain activity. So we have different methods: single cell recording, MEA, ECOG, EGG, fluorescence imaging type, etc. So we'll deal with the individual one. And when we talk about techniques that we are using to study brain structure and function, because the brain is actually the center of human body, which actually sends and receives millions of signals every second, day and night, right? So even in the form of hormones, nerve impulses, chemical messengers, the impulses would be moving back and forth, the body to the brain, brain to the body. So this exchange of information makes us move, eat, sleep, and even now, right now, you're thinking about, what topic has to be covered. So these are all taken care by the brain. So obstructions such as tumors can interrupt normal brain activity. So if there is any tumor within the brain regions, so that would hamper the brain activity. So we call it as the tumors which are growing, which actually has the capability of blocking the signals or leading to deficits of normal reasoning and motor controls, movements, etc., or even consciousness. So these will be affected if there is a tumor within the brain. Since the brain is extremely fragile and difficult to access without risking further damage, imaging techniques has developed very well in past decades, so that we can actually visualize the brain structure and activity by using different methods. So, per se, let's see an outline. So what are all the methods that we are going to cover in this, video? So first one is examining the tissue. So tissue, as I already told you, need to remember this: cells forms tissues, forms organs, combinedly to form organ system. And organ systems combine to form organism. So we are talking at cellular level at the same. We are talking about tissue level also. So examining the tissue would be one of the methods to study the brain activity, etc. So here, if you see this tissue, right? So first one, we are speaking about first one. Yeah. So the options for measurement of brain structure were effectively limited to postmortem. So actually, in a living condition, previously it is not possible to study the brain structures. So it is limited to postmortem. So after the death, we will be studying the brain tissue, and very in rare occasions we use biopsy methods. So we exercise certain tissue and we'll try to see. And there is later part, there was a drastic change in the techniques which involving the removal and analysis of small samples of brain tissue from the appropriate area. So in rare instances and because of the advancement, we are able to examine appropriate areas which it belongs to, and we can actually talk about how this brain functions are happening. So for that matter, in any case, if the external appearance of the brain at postmortem may be entirely actually normal, with damage or disease only apparent on closer inspection of internal structure of the tissues. So even after the death, like if we see the brain, it would be very normal looking brain, but if you see the tissue level only, we will be able to see that what are the problem areas or what are the issues that were there in the the brain after closer inspection of internal structures of the tissues. So actually brain tissue looks solid to naked eye. So investigations avoid technological developments as well. So in gradual way of refinement over many years, the light microscope have shown a lot of degree of inventions and discovery of tissue staining techniques, which had a tremendous effect on highlighting particular component structures of the tissue. So if you take a tissue, we can actually use certain staining procedures so that we can visualize in the light microscope with the use of light microscope. So right now, light microscope can now reliably magnifies by several hundred, but electron microscope can magnify by factor of several thousands. So now microscopy has been advanced. First, we used to have a simple microscope. Antonio van Leeuwenhoek, who actually invented with this microscopes, lenses, etc. And later on it developed, and the microscopy. So the microscopy, which came a long way from simple microscope to the light microscope to the bright microscope, the confocal microscope, then and then electron microscopes, both TEM and SEM. Transmission electron microscope and scanning electron microscope. So these are advancement in the microscopy led to the study of these tissues very much, and especially they can magnify factors of several thousands, and even they can produce images of tissues or individual synapses. So synapses, till synapses we can actually study, or surface of the neuron also you can study by using this SEM or TEM. So this is how we examine tissues, and we use different staining methods as well. So when we talk about staining methods, so we have silver staining method, or HRP, horse radish peroxidase method, which is most reliable staining techniques. So even the scientists would use the combination of silver staining method and the HRP techniques, which establish functional connectivity between the brain regions. So these are two brain regions. So this connectivity we can explore by using silver as well as HRP staining techniques. So this is about examining the tissue. How we could examine, either we can do the biopsy in rarest cases, and we can study after the postmortem, and we will be studying very clearly about the parts of tissues, and we can use light microscope and microscope. Advancement was seen and in past decade, and right now we can use scanning electron microscope to get the surface details of the neurons or synapses, and we can use the silver staining method or horse radish peroxidase for that matter. HRP means horseradish peroxidase. Horseradish peroxidase. So now let's see the second, so that is nothing but lesions and ablations. So the second technique we can use is lesions and ablation. So what it means, lesions by cutting the tissue. Ablation is nothing but removal of the brain tissue. So these techniques are used by neurology as discovered effects of behavior of lesions and ablations. So we can, by doing this method, we can understand the behavioral changes as well. So outside output as a behavior, we will study that lesions in some areas can alter the behavior of the human. So initially, there would be a period of orientation during the time. If you see this lab animals like mice were subjected to certain training and orientation, and then they will be looking into the behavior, whether the same behavior is shown after lesion or ablation. So, for example, if the mice locates the maze, around the maze, if it can locate a food pellet, then the scientist would remove a small region of the cortex, and following period of recovery, we'll see many trails that either animal is relearning to understand the maze and finding the food pellet. So maze is typically a sort of puzzle, like there would be an entry point, and there would be different directions that can reach the food. So after removal or cutting, they will be seeing whether the capacity to relearn the maze is there or not. So these studies are done by Lashley, and but in the lab animals, and of course for specific regions or obvious reasons, the procedures are not actually experimentally used on the humans. So we haven't used on the humans. But when there is a patient with tumors where that is a possible, operations got done, and the study of these areas also by lesion and removal of certain part because of the cancer or because of the tumors in the brain, that showed a path to understand the human brain. And it also includes lesions by application of chemicals or drugs. So this is about lesions and ablations. So we'll now talk about the other one is electrical stimulation.